Hello, my name is Zahra Sohaila Sohaili. I am an academic member at National Institute of Genetic Engineering and Biotechnology, Iran. Today, I would like to share with you one of my recent papers entitled miR-96, a promising target to ameliorate the symptoms of diabetic retinopathy. miR-96 is overexpressed in the serum and retina of subjects with diabetes. We investigated the functional role of miR-96 in INS-ACT GLUT4 signaling axis, which is the main pathway for the transport of glucose in cells. Gene expression levels of miR-96 and its target genes were measured in vitro under high glucose condition in the retina of streptozotocin-induced diabetic mice, in the retina of AAV2 miR-96 received mice, and in the retina of human donors with diabetic retinopathy. MTT assay, bone healing test, tube formation assay, Western blot, tunnel test, and angiogenesis assay in vitro and in the examined retinal sections were performed. Under high glucose conditions, miR-96 expression was increased in the mouse retinal pigment epithelial cells in the retina of mice received AAV2 carrying miR-96 and STZ-treated mice. Following miR-96 overexpression, retinal pigment epithelial cells proliferation decreased while the cell's motility was increased. UVEC cells treated with conditioned media of miR-96 overexpressing RP cells showed increased tube formation. Western blot results revealed the expression of FOXO1 and FOXO3 proteins increased and expression of ACT1, PDK2, and PIK3R1 proteins decreased in miR-96 treated RP cells. Histological analysis of the retina from STZ-induced diabetic mice model and AAV2 bearing miR-96 injected mice showed that total vessel length of the diabetic group was increased as compared to the non-diabetic controls. INL, IRL, and whole retinal thickness in diabetic mice and AAV2 miR-96 treated mice were significantly decreased. Tunnel positive cells were detected in the GCL, INL, and ONL layers of miR-96 injected mice and in the GCL and INL layers of diabetic mice. Gene expression analysis in retinal pigment epithelial cells under high glucose condition and or miR-96 overexpression, STZ-induced diabetic mice model, AAV2 miR-96 injected mice, and human diabetic retinal samples revealed that miR-96 regulated the expression of several genes in the INS-ACT axis and some genes involved in GLUT4 trafficking, more or less in the same manner. Since disruption of the INS-ACT GLUT4 signaling axis causes advanced glycation, end product accumulation, and inflammatory responses, inhibition of miR-96 expression would ameliorate diabetic retinopathy complications. I have uploaded the article for those interested in more detailed information. Thank you very much for your listening.